that belongs to Maharashtra and especially lies in Western Ghats, and this is administered by Aurangabad district as well as Jalgaon district. Next please. Then this is the area which is covered by this Gautala sanctuary, that is around 26,061 hectares of 64,399 acres, and that is considered as a reserve forest area. Now you will find that this area is called as a Gautala sanctuary because of some mythological states. That is said that Gautam Rishi belongs to that particular area, or that he passes from that area, and that is mentioned in Ramanathapuram, and then it's considered, that area is considered as a Gautala or Gautani or Gauta. Then many communities that are dependent in that particular area, and they utilize that particular plants that are present in that particular Gautala sanctuary. Not only we find that indigenous communities that are present in the Gautala sanctuaries, that are Hivi, Marathi, Ammal, Halal, etc. These communities, they are living in particular towns, particular villages, particular villages, and scattered in that particular Gautala sanctuary area. That people are very simple. The maximum, if they know about the word, so that Kannada Taluka Vekhi Basa. Apart from they don't know the district area or any other part of the world, and their survival is up to only that particular region. They utilize plant for everything that we will find. This is the one community, that is the Ammal community. One of my research scholars is sitting with them, and you will find that their standard of living is very simple. Whatever the natural sources are there, they prepare their houses with the help of that sources, and whatever their house, that simple house contain everything. When we go inside, so we will find that they are having a simple big room. That room is considered as a guest room, kitchen room, drying room, store room, as well as bedroom. And they survive in that particular standard, that particular methodology and that particular standard of living. But they utilize the plant and they protect them. That is the important part. This is the Thakur communities. You will find that they are having good culture. Every day they used to utilize whatever the natural sources are there. They prepare ropes, they prepare carpentry items very nicely. Whatever the plants are there, they utilize for different treatments, as well as they also having a good humanity. When we go over there, they use a lot of respect. And whatever is there, information is there in their heart, they used to provide that particular information. So this Thakur community is mostly present in Thakurwadi area, that is a particularly known area. And in that area, we will find that these Thakur communities are present. Then another community is Bhil community. This Bhil community is also having their own faith, own tradition, slightly separate from the Thakur and Banjara and other communities. And they even utilize whatever the things are there. If there are certain religious spots, so they don't utilize, they don't take that particular plant or plant species from there. although in Gautala century there is no any type of sacred groves, although that area is having a lot of religious significance, but sacred groves are not there. But local people, whatever the plant they require, they take from the forest, not from the temple area, not from the religious spot, but they utilize from some other sources. So in that particular area, we will find that these communities are present. The next is Pandi community, very famous community it is. They normally use stone carving, and they entirely the most important target at present is utility of the plant for the human being. So they utilize that plants for different treatments, for different cares, and here you will find that they are utilizing the plant species in particular region. This is another community that is present in Gautala century. Next is, then the most popular community is Banjara community. This Banjara community is widely spread not only in Gautala century but also in Kannada forest or Aurangabad district. And even you will find that this Banjara community is present in different parts of the world, and in our adjacent countries you will find that they are surviving over there. They originated from that particular site. Then they are having their own culture, own tradition. Next please. And we will find that their costumes, their identity, especially female identity, is most important one, and they preserve, even though many of the community persons they have got many good posts in government offices, but even in that particular level they preserve their cultures. So we will find next please. These are the communities which you will find that their traditions, their dances, and many other things you will find over there. Next please. These are these are the qualified ladies, and they are preserving their culture in a particular manner. Next one. Now, what is the methodology of that particular research? Actually, these communities are having the informations about the utility of the plants. They know very well about that particular plant. They used to collect that particular plant and utilize. Apart from that, they are having the local name of that particular plant. And they also knows where these plants are available. Then after that one goes administration. That is also the most important one. And even conservation of that particular plants. Many of the peoples, they take honey from the forest, but they never kill entire beehive. They take 75% of that bee, they cut 75% of beehive, obtain honey and sell in the market, in the market, in very reasonable rate, and that too in original manner. They don't know about the mixing or adulteration. They don't have knowledge about that. So I used to go over there, collect the information, used to visit in forest with them, collected a specimen, brought that specimen in the laboratory, identified with the help of floras, local floras, generalized floras, and some of the species I have tested in the laboratories and confirmed the utility of the plants and presented it in different research papers. So this is the first important thing. Then that local peoples, elder peoples, that are the sources of this information, and especially the persons, those who provide this medicinal information, is considered as a shaman, bhagalas, vaidus, kunjalas, not only Microbiology used to call it as a medicinal man or medicinal female. Next, please. Then these surveys are made in different patches of different small villages of Kannada area, local assembly area, stayed with them, enjoyed with them, involved in their cultures. That is a very interesting part. They used to dance every day, and I have participated with them, and then they share their information. Next one. Then some of the plant species I am presenting over here, apart from the main, almost 250 plant species, mostly in that particular areas are present. I have collected them, and most of that utility is having their own utility. Confirm that utility with different literature, different researchers work. Out of that 250, 150 plants, what I observed, they utilize for ethno-veterinary purpose. Means they also utilize these plants for the treatment of animals in various ways. That also I presented over here. Some of the plant species that I am showing here with the slides, that is Ammal sculentus, locally called it as a handy. Normally, the local people utilize for different diseases, for fever, treatment, dysentery problems, or urine infections. They also utilize this handy, that is sculentus, for treatment of animals. They utilize the leaves, dried leaves, and they tie that dried leaves around the swelling part of the animals. Ultimately, it controls that paste of the fruits, that also utilize in with hot water for removal of placenta after parturition. And this this body is having again belongs to Malvaceae family. It's having a fibrous structure. So they take that plant in green condition and obtain the fiber and makes beautiful ropes. I have taken the photograph of that ropes. I will show you later on. Next, please. Then this is called as a plant name or wild lady finger or gully lady. That is called as a Ammal manica. This plant is also available in large quantity in that century, and that decoction of the root that local people utilize for digestive problems as well as some other problems also. That bark fiber is useful for preparing ropes also. Fine ropes they used to prepare with the help of their fingers, and this is the great technique, great knowledge they obtain. Next, please. Then Ammal precatorius. This is very common plant, available everywhere. It is called as a bunch. If you are able to see that bunch, so you will find that the local people, especially the seed paste, is useful for the treatment of paralysis, infertility, abortion, and the leaves, that is very common everywhere nowadays, that is useful for treatment of throat trouble, cold, cough, etc. If you people are familiar, then this leaf of the gourd will be added in pan for shaving purpose, and the important reason that it solves the problem of throats or throat infections. That's why it's very common, but local people utilize as a treatment. For animal treatment, even they utilize this seed mixed with coconut eye and used for wounds, or that roots of this one is useful for ulcer treatment of animals. Then this is a very common one, that seeds is used for weighing ornaments or gold in jewelry shops. That is the very common one. Next one, please. This Abitulan indicum. This is very common. It is called as a mudra, or you will find that stamp-like structure is that one. That's why it's called as a locally mudra or patari or shika kabad. That Abitulan indicum is also having a lot of medicinal significance with the local people. Normally, they used to take the skin, that leaf, prepare that paste and utilize that paste for skin diseases, infections, itching and other purpose. And I have seen personally that in a short period, whatever the itching is there, infection is there, that comes under control. Then we also find that this leaf of paste is also useful for making the decoction, and the decoction is useful for mouth problems, bronchitis, diarrhea and fever. They used to give this decoction their children for controlling their fever. Apart from this one, next previous one please. This mudra is also having utility for animal treatment purpose, and they utilize this leaves for eye diseases of animals, and leaf paste is also useful for various infections. As we know that animals were attacked by certain insects, certain mice. So at that time they put this leaf paste or leaf drops on that. So whatever the insects are there, they removes from the body. And this stem fiber is also useful for preparation of ropes. Next please. Then acacia catechu, very common, car, tatha, hair. Now this plant in nowadays in endangered condition, only few species are left over there. Otherwise, majority of the plant species were cut by certain other persons, or can't say that one. But this gath and katham is which normally obtained from the wood, central part of the wood. We got it as a gath, and that used in pan for various purposes. It gives red color. This is useful for bleeding piles, uterine hemorrhage, leukorrhea, glit tincture, and even many female problems, mammary gland, pain, and many other things. This garlic is very commonly used. Then veterinary will find that this bark is useful for wounds of treatment of wounds of animals, or controlling the cracks of other animals, and that is used for the parrot and diarrhea also. Then this scar or the acacia get into root part, which is considered as the hardwood of the plant, is used for making the agricultural instruments. Wood pieces boiled and used in metal leaf as a bark, and that central part is also useful for coloration of skin, and that is also useful for tanning industry. Apart from this one, this scar is also used for dramas or movies for coloration like mehndi or some other makeup. This liquid is also very useful, and that people know very well those who are on the dramas or prepare the films for that purpose. It is also useful in their many of the purposes. Next one, then Asian Sura. This is considered as a large hair. This Acacia Sura is also having a lot of significance, and useful for cough, mouth, and throat infections, as well as certain pulmonary problems. Even it is useful very commonly. Generally, it is useful for dry purposes because lal hai, red hai is the name, and that is useful for dye purpose clothing. Especially, it is used in dyes and medicinal industry, and useful for beans, as well as the wood is very hard, so ultimately it is useful for making agricultural equipments, the instruments that were the help of wood. So local people utilize its wood, and the bark waste is useful for treatment of animal in thinness, or it is called as emaciation, that is useful for unwanted fat control of animals. Next please. Then acacia fibulina. This is called as a pandra, very white hair, and commonly called as a hair. These are two trees. You will find these trees in almost all the part. But in Gautam century, the bark decoction is useful for urinary treatment, urinary problems, especially in male, and this hardwood is used for making the carts as well as agricultural implements. And if they utilize this one, then whatever the wood remain out there, they utilize it as a fuel purpose. In animal treatment, stem bark is also used for treatment of diarrhea. So this white hair is also having a lot of significance for various treatments as well as controlling of many problems of human being as well as animal, and even it provide lot of economical source. Next, this acacia leucophilia, locally called as a river or river. Red we have seen, white we have seen. This is green one. This is also very common, and the bark is having the same type of utilities, that is for bronchitis, bilineus, ulcer, and many other problems they utilize. Veterinary, it is useful for dislocation of bone. Actually, bones they take a small piece of this green bone or a bone, cut it, and if there is any type of dislocation in animal, so they used to prepare with the help of normal tradition and tie it around it. So whatever the dislocation is there, after some time it becomes normal. Then generally it is useful for making the toys and forming instruments. Bark is used in preparation of local wine, that is called as a country wine, that they are having a term locally, Deshiyaru, that is called as a country wine. So locally they use to prepare that one and utilize that one. One important that one, that if this acacia area is growing in a group in some one area, the local people say that the water level is very high, and if you take pouring water or want to open dig a well, so that area is very particular. On the basis of the population of this one, the local people are in our area. There is a lot of scarcity of water. In some area, large quantity of water is available, but in many rural area, no water is there. So at this time, they used to take the pouring from the earth or dig a well. For that purpose, the quantity of water gives a lot of importance. And if there is not getting any type of water, so it is lost, very totally lost. So on the basis of this population, the local people say that the good water level is here, and if you want to dig, you can dig a good water. So this is also a somewhat use of this particular plant. Next Acacia nilotica. This Acacia nilotica is again very common plant and grows everywhere in the area where we will find that this plant shows lot of significance with reference to human health. It is useful for controlling diarrhea, dysentery, vomiting. Barley is also useful for mouth cleaning teeth. Later on I have seen in different regions the people used to prepare toothbrush of this one and it implies for cleaning the teeth. So that is also common. In some areas I have seen that this local people drive, prepare coal, just make it powder and used as a tooth powder of this one. Because it is having a lot of significance about the entire effect of the mouth. Then in animal treatment, local people utilize the branches of the leaves and especially reports that use cattle and sheep and goats. It is considered that if that goats or sheep or whatever the living animals are there, if they utilize this one, so they use milk in large quantity. So this is another significance. Apart from this one, it is useful for making agricultural implementation, then making huts, homes, useful for making the appliances, especially domestic kitchen appliances, and bark is used in tanning and dyeing industries. It is also very common. Next one is then apart from this one, society of large quantity of plant trees there. And if you go in details, we will find that this utility is large one. Here I have given one, the basis of the sum of plants, and you are able to see. So you will find that these plants are having a lot of significance. They will utilize in different manner. Their mom, tea, cacao, banjara, they utilize that this is not very common. Everyone utilizes for various purposes and almost in all treatment they utilize. So due to this utilization, what has happened? The exploitation of the population of this I know that plants is in rare condition. Some gardens, some people are surviving over there. We are also throwing garden for conservation of this particular plant. And local people, they conserve, but outsiders they are coming, they are destroying this one. So what's happened? These communities nowadays they don't allow these outsiders to come inside that particular area, and they are conserving these plants in that particular area, whatever the fields are there. And if someone from the outsider comes over there, so they used to take the interview, they ask them, and if they require any help, they provide. And if you want the plant species, if that plant species are available in large quantity, they themselves use. But now that they are propagating this particular plant, they are conserving in large quantity. Then eagle marvelous is also in that one. Please, next one. These are some other plants that are very common in our areas also. Arabian salvifolium is surviving only on 2 places over there. They are conserving it. Now, I hope that it will propagate again. Albizia also there. Then next one. These plants useful normally the local people, especially the Thakur people, useful for making the fibers. This I will must say Bombay Siva, Catechorn, very common plant having beautiful flowers, Beautiamonas keratopus, white plants, erythranum, and that Pergularia. These plants normally that people utilize and they conserve over there. they obtain the bark, they take the required part from the plant, they protect over there for again getting the new bark or new species or in next year they again obtain that one. They also take the seeds and conserve, they grow sows inside the soil and conserve in that particular manner. Then for making household implements, that local people utilize these particular plants that we are seeing over here. Dendroclamus sticulus is very common over there, and many other plants are there. They utilize, and this is for carpentry, for economical purpose. They utilize for making different furniture items as well as domestic required item in that particular area. They utilize this particular plant species. And pulse preserver is also there. Tectona and WGSC rarely present in some of the patches of that our Gotala area. The next one is even they are having their faith on the religious purpose. So they utilize that plant for their different rituals, religious rituals, for puja purpose, and these plants for that purpose, for that religious purpose, they are conserving over there, they are growing over there. Just see that city persons used to purchase the material and offers whatever their faith is there. But this local purchase, local people, purchasing is considered as a bad habit. So what they used to do, they used to grow that plant, they conserve that plant, and whatever the sources is there, they utilize and offer their feelings, their faith to that particular area or gods. So this is just somewhat brief information related with the ethnonautical utility of Gautala century. And apart from this one, there are many research work, especially with reference to particular diseases, particular animal treatment, as well as particular social, religious, and what you can say that utility of the plant for various purpose. Some of my students, they obtained a particular, they constructed a community like Banjara community. They perform their research work on that community. Another on Thakur, another on Game. Such type of research work is going on. A brief information I have presented for here. And if you feel send it out any difficulty, any confusion, so you may ask. Yes, sir.
They cause so many disadvantages to our physiology and our biochemistry. So why not still that these medicines prepared from particular thing? So for all the diseases, we are having the possibility of preparing medicines only from the medicinal plant. Why these particular field has not developed as we expected? So that is
Yes, sir. This is a good question. Although less side effects are there for the utility of the plant. Normally when we present such type of informations among the youngsters, so at that time we used to say that this is the utility of the plant. But don't take any type of medicine directly. Taking it directly means you are utilizing the plants. And how much you are supposed to utilize that plant. What is your dose administration? For that purpose, you have to take the opinion from them experts. The experts use the dose administration that you take 1 seed or 2 seed or this much water or this that much. Why? Because of your health, your age, your resistance power, and then it becomes normal. It avoids side effects. What's happening, sir? After taking this one, some of the medicines they use their effect in a long time, a long period. But allopathy medicines, they use quickly their response to that effect. That's why people is going over there, apart from the many type of side effects are there. This utility of the plants is also becoming more popular. Why? Because of less side effects. And I hope in future the many people they will utilize this plant in a particular manner, and as per your confusion that whatever the utility is, that becomes more popular.
Anything else?
Anyone?
Sir, thank you, sir. That was wonderful and interesting. Thank you. Now go to the session. You are sharing the session, sir. We are having a question in the session.
Yes.
We are
So
V1016 gene mutations could contribute the observed systems, although we have to identify more resistance genes in them. And then identified resistant genes, we have to sequence whether it is particular gene or not. So we are about to giving the samples, positive samples for sequencing. Thank you.
Any question?
Yes, sir.
Because these some of the hospitals which are having the resistant power of that pesticide that we have used, isn't it? But this is very common for especially for the very small organisms. Even we are continuously changing the use of that pesticides, isn't it?
Yes, sir.
Previously, for controlling the mosquitoes, we have used only for spraying the DDT.
We have to monitor the target whether it is suppressing or developing their cells. Right? Thank you, sir. Thank you, sir.
The two pipelines are administered by the local bodies.
Sir, sir.
The two pipelines are cited for your
No,
study.
as per the suggestion, ICMR, we have done this.
The chemicals are used by the local.
Yeah, yes, sir. Yes, sir.
Control must be.
Yes, sir. Yes, sir. Thank you, sir.
Ma'am, simple.
Yes, sir.
You say for specific mutation.
Yes, sir.
This mutation is already reported, right?
Yes, sir.
How
No,
to say there is a prominent mutation or pesticide this mosquito in a second?
actually 2-3 genes are there doing, sir. This is one of the genes here. Some other genes also my senior reading very sample papers that we have published, and one more genes are doing. Not only one gene here. Thank you, sir. Yes, sir. Please, sir.
Very good. Nice presentation.
Thank you.
Thank you, madam. Thank you.
Sir, I'm asking.
Asking question.
Good morning, ma'am. Sir, I'm coming from Chennai. I'm an entomologist. I just have one question for you. How far your mutation works for reproduction? When they lay eggs, how far your mutation or your
It will take over, like transfer and transmission is
does happen here?
Yeah, it is happening. F1 generation also we are doing. Not only field cat mosquitoes. F1 generation also we are doing, and it is literally the same system.
So,
We most probably doing F1 generation.
our work is that we don't want mosquitoes generation, like how they
She's asking about the egg
egg capacity and mosquitoes.
whether the mutated mosquitoes
Can
egg
does
capacity
it affect the egg laying process of an
No,
it doesn't.
no.
Then how come you can control the
Yes, ma'am.
generation?
For that only we are doing the 6 months monitoring. Every 6 months we are giving the proper report from local populations. So according to that, we have to identify the target site. So every 6 months we are giving not only one gene we are doing, another gene mutations we are finding out that. So we have to identify the target site. That is more important. So based on the target site, we have to improve the insecticide power and we have to suppress it.
We can an area for study come for where is my area? Can you do any kind of research like this in general?
No.
Do you have any idea?
I'm doing actually. Continuously I'm doing.
You can collaborate.
Yeah, you can collaborate with me. Can I?
Yeah, sure.
Okay, if like study you can do it in Chennai.
Yeah.
So we will arrange everything for you.
Thank you.
Thank you, ma'am. Thank you.
This is the achievement of
Yes.
Thank you.
Next, the recognition. Good morning. Myself, Ramanathapuram. So my topic is on habitation of bioactive polymerase matrix based on Tylosin and Urate. non-derived human nanoparticles for moving in that application. So I'm from the Department of Zoology, Guru Nanak College. So moving on to the introduction. So the basic introduction for this topic is how are we managing the crustacean waste. So around 6 to 8 million tons of waste each year is being not properly managed. So through the crustacean waste, lots of byproducts are made. So among that, crustacean waste have made the byproduct, that is the chitosan, have taken the chitosan from the crustacean. The crustacean have chosen as the poisonous sand limiters, that is crab. Next. So what is the property of chitosan? Chitosan, it's a multi-phases application. It has healing capacity, it facilitates filtration and healing, then cardiovascular health, and it reduces the cholesterol and the lipid levels and antibacterial properties as well. Also, it has its use of the hydrogen. They have hydrogen composition, and then antioxidant properties, tissue regeneration, supports cell adhesion and preservation. Then there is cell adhesion, others species from another cell, and it interacts with the single pathways as well. Next. So this is the chemicals and methods. So first, the crab cell is made, it is broken into smaller pieces, and then after the pieces are stages, there is the nitration, mineralization, and also we have the acetylation. The process where the chitosan is prepared. So once chitosan is extracted, moving on to the sister nanoparticle synthesis. So sister nanoparticle synthesis is usually in the ranges between 400 to 500 range. So I got around 4.3 nanometer with the help of the UV spectrophotometer analysis. And then moving on to the characterization, I've used different instruments, that is Fourier transfer, infrared analysis, that is FTIR, and then EDAX, then TEM and SEM, and also ELS potential, ELS, and zeta potential. Next, so these are the techniques I've used, that is the UV for monitoring the silver ions, to measure the light absorption rates of nanoparticles, and FTIR to identify the groups, that is the functional groups. It can be aldehyde, ketones, or alkynes and alkanes. Then SEM and TEM to know the morphology of the samples, and then DLS to assess the particle size and polyviscosity index, and then the zeta potential to determine the surface potential of the particles. Results and discussion. So here, if you see in the first picture, this is the pie chart that shows the swelling capacity of the chitosan hydrogen. So mainly, the chitosan hydrogen has to have this property, because when a chitosan hydrogen swellens up by absorbing a lot of water, it creates a good environment for the cells to occur. And if you see, I've used three different solutions, that is PBS, that is phosphate glucosaline, and methanol and water. So among these three, water shows the maximum amount of swelling, that is around 49.4%. Comparatively, it is greater than other solvents, and that shows that promotes the moist environment for the cells to occur. Then the next picture shows the antibacterial activity of silver nanoparticles. So I have taken E. coli, Staphylococcus aureus, Pseudomonas aeruginosa, and Bacillus subtilis. So why specifically E. coli, Staphylococcus and all these things are? They are the major ones which is causing a wound infecting pathogens. They are the major wound infecting pathogens. So E. coli and Staphylococcus aureus are the most common wound infecting pathogens. So in E. coli, it showed at 100 ml concentration, it showed 23 mm of inhibition and 20 mm of inhibition, and in the control it showed 23 mm of inhibition. The control, the antibiotic used here is the gentamicin. The Staphylococcus aureus, it showed 12 mm of inhibition, which is lesser. Comparatively, you see Pseudomonas aeruginosa, it showed 15 mm of inhibition when compared to the control. The third picture shows the antimicrobial efficacy of the chitosan hydrogen versus chitosan hydrogen with silver nanoparticles. So the red spot is the chitosan hydrogen with silver nanoparticles. The E. coli showed 11 mm and the next the highest one is the Staphylococcus aureus, which showed 8 mm of inhibition. And the next one is the Aspergillus and Candida albicans, these are the fungus. So the antifungal activity showed 8 mm of inhibition in Candida albicans. So this is the outline of all the antibacterial, antimicrobial activity. Next. Characterization. So this is the 10 and 7 observation. So this picture shows the 7 observation. So A is the particle magnification. The next one is the chitosan hydrogen. So you see the chitosan hydrogen main purpose is the it has a property of porous structure. So porous structure basically helps in the permeability of the nutrients or but it is impermeable to the bacteria or any microbial activities. The next picture shows that the C is the 10 ppm of hydrogen hydrogen to silver nanoparticle. So silver nanoparticle is a crystalline nature. So if you see in the picture, it is embedded properly to the hydrogen hydrogen, and hydrogen hydrogen has the silver nanoparticle in the crystalline. Next is the 10. 10 shows the particle size and the measurements it is shown, and it is embedded to the hydrogen hydrogen silver nanoparticle. Next. In the two studies, so silver nanoparticles, nanoparticle. So of course, silver nanoparticles or the metallic nanoparticles has a very toxic. So at the 72nd hours, they have taken a time limit of 24 hours, 48 hours, and 72nd hour. So at 72nd hour, it showed family is perfectly with cell biology, and then the hydrogen it showed 85%, and the hydrogen silver nanoparticle it showed 86% of cell biology. So comparatively, hydrogen with silver nanoparticle has a good cell biology. This shows that the cells can other and they can migrate and form new cells and we can proliferate this. Next is the scratch of cell wound healing activity against the L1 cell line, that is the government L1 line. So here in the carbon you see the control and the hydrogen with silver nanoparticles, so it is against the healing of the cell. So on a day-to-day life, we come across a lot of wounds, like burn wounds, or so wound is something a part of the life. So we know how we have the experience of it. So it is a traumatic experience for people who have a chronic wound. So it is better to reduce the traumatic cases and we can enhance these kind of points given only a topical application. So it's not a concentration method, but given a topical application. So like an ordinary ones we can use for the burning itself. So this shows that has a maximum amount of healing capacity because it regenerates and enhances the quantity within bundles of collagen that will promote the damage cells to repair. So this is the picture that shows previous slide that showed the carbon picture. So the silver cover showed the silver cover is the control and 77 cover you see there's a small gap and that's where it is not happened because the group has not properly healed. But at the 72nd hour of the virus and hydrogen with the silver nanoparticle it showed the maximum number of amyloid cells. That is, the cells are viable and they have formed the cells of body and that shows that the wound has been healed in the cell. So the approach study remained more development like an additional variable to enhance wound healing application that combines hydrogen and silver nanoparticles which is a natural bad and plant species. So with the facility that combine antibiotic properties and mix it with the hydrogen matrix to form stable mixture. So according to microbiology testing, the common infected pathogens are respiratory and stable also obvious. And then the hydrogen hydrogen exhibits the biocompatibility, reflects in its maintaining cell viability over 95%, and it can be used as a wound healing material. The hydrogen hydrogen is a nanoparticle group is the most effective wound healing, well-known structure dense, collagen composition, and reappearance scale appendages, confirming the synergistic effect of hydrogen and silver nanoparticles.
Presentation. Basically, basically,
They have the wound healing and scaling, but comparatively.
already hydrogen also included in the graphics as well as all the other
Yes.
hydrogens cell.
Yes.
It says 95% the performance in the nanoparticle and hydrogen induce proliferation of your cell. What way to proliferate the hydrogen to the weight cells?
Yes, hydrogen itself has the wound healing ability. So basic idea is to infuse 4 different properties. The single nanoparticle main property is when there is fused with fibers of hydrogen, its property is enhanced to have the air capacity. But in single, as well as in single nanoparticles, the major property of single nanoparticle is anti-microbial. So when I am using these two properties, I am using it together. Each one has their own property, and they exhibit to enhance the cell viability in the inhibition. That is the main concept.
Cells is normal cells are cancer cells.
This is normal term. We need the and also even you study a red ball as well.
But your cells like your cytosol have the ability to mitogenicate, right?
Microgeria. Yes.
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 Certainly have mitogenicate.
You have data for hydrogen separately and single nanoparticle separately and combined both. You have data. Only data.
Only activity has shown in the graphs.
You just mentioned control and experimental. In that, you separately hydrogen and single nanoparticle only cavity. Here, you have data until the combined.
For this, the combination is actually only so specifically the previous cycle also we had a separate sample.
What is the control group? You mentioned
I'm not doing anything.
very frequently.
It is normal only habit. There is no natural.
Right. So, hydrogen has only capacity separately. Then single nanoparticles also having the only capacity. Yes, right. So you combine both. So how I enhance so separately you have to do hydrogen and other single nanoparticles.
I've also done biostudy in a separate mechanism. So this is for in vitro study.
When you combine the effect of hydrogen and single nanoparticle,
Single nanoparticle.
single nanoparticle is better than
Titles of hydrogen and titles of hydrogen and single nanoparticle are more
around 90% survival, right? Okay.
Satya
instrument
Good morning, all. I am Satya, research scholar from Central University of Tamil Nadu. The title is Vector and Rodent Surveillance Revealing Molecular Evidence of Polycarbonate Delta District, Tamil Nadu. Next. So we all know Staphylococcus disease that is caused by this mite called chiggers. So the larger base of the mite called chiggers cause this disease. So this mite harbors this bacteria called Organised that is responsible for this disease. This Staphylococcus is not dependent on the bite of rodent. These mites are present on the pinna of the rodents, from where these mites they harbor this bacteria. They transmit this disease called Staphylococcus. So this Staphylococcus is one of the ligaceal diseases. There are other diseases caused by the ectoparasites present on the rodents themselves. Okay, so the rationale of my study is actually the study area is Tiruvarur district, which is one of the Kaveri Delta district of Tamil Nadu. One among them. So this, it is a rice bowl actually. Tanjavur is called rice bowl. Here, the major occupation is agriculture, and we were able to see more packing into rodents there. So we, when we hear the farmers make some so much trouble because of the rodents loiter in the places, and they eat all the fields over there. So
that is one among the concerns that we chose this Pirwara district. The research gap there is the absence of ectoparasite surveillance. As in, there is no entomological surveillance in that particular area related to the Streptococcus vectors. Also, molecular identification and circulating pathogens in the trigger mites is also lacking. Inadequate understanding of vectors of Streptococcus and also other pathogens like Leptospirosis prevalence in rodent hosts. So these areas are the place where we have to explore more. So that is the significance of this study. And my study addresses this critical knowledge gap in the ectoparasite as well as the rodent in the needs of surveillance for this both Streptococcus and Leptospirosis. And also it enhances understanding of vector-host pathogen dynamics to guide public health interventions. And also it generates the baseline data for the geographic distribution, that we get to know what are all the places where we find these Streptococcus cases more. Next slide. Yeah, so this is one part of my PhD. So I have taken two of the objectives, like entomological surveillance and molecular characterization. So entomological surveillance entitled, so we have to do whole district surveillance. We will place traps and collect rodents like that the study goes. Next. This is the Pirwara district. Almost all the blocks of Pirwara districts were covered. And there are two different traps for catching rodents. Okay, so there is this Sherman and Button trap. So there are many difficulty in catching rodent and also taking to the laboratory and check for observe for the presence of ectoparasites present on them. So like in the around 5 p.m. in the evening, I will send the traps and the traps will be checked next day morning. So the presence of rodents, if rodent like rats, shoes, whatever is present, we have to, I will take back to the laboratory and will observe for the presence. So this one I have already done. I have observed for the presence of ectoparasites like ticks, mites, fleas all over their body. Next. So this is the triggers that cause this particular Streptococcus disease. So this is the winner of the rodent, where these triggers are very tiny, around 0.2 to 0.5 mm only. And they are very pathogenic also. If the bacteria strain is present from them, they move fast from the ears. So we have to be very careful. We have to anesthetize this rodent. That's how the study works. And then these triggers, once rodent blood circulation is clogged, like as in the rodent has is losing its life, in that case what happens, this triggers tries to move outside the body because the sucking of the blood is it is difficult. It will not get enough food, like whatever tissue fluids it will get. So it tends to move away from the place where it is located. So as soon as we anesthetize them, it is our responsibility to check for the with the brush. I usually take this trigger from the vena, and in the body part is fleas and ticks will be present. That also we have to observe by combing it and finding the species. These species, whatever I have collected, I have done molecular morphological identification before confirming to the molecular. Next. So there are many taxonomical keys book available to identify these trigger species. There are around 3,000 trigger species. Like you can see the previous slide. Triggers are very tiny. By the naked eye, it will be look very small dots. But each trigger varies based on the sputum and other mouth parts that they have. So you imagine 3,000 species is such a big difference that these taxonomists have already segregated them and they have identified based on the fetal pathway. So many identification keys, taxonomical keys are there. So
we may get a new species that we have not identified at all. We have got 6 new species, but with the help of ICMR, DCIC field station Madurai, I am trying to figure out what it is that. And one thing now we can see here, adult also I got to find in that. Still be there in the soil only the garbage will be moving on to the ears. Yeah, next. So this is the sputum. Once after getting the chigars, I will put it in the mounting line and clear the contents what are there inside the chigars, that are the internal tissues I have to remove, and then I have to mount it in the slide and visualize under the microscope. There are several specific characteristics which we can easily identify. That is this sputum. So this sputum is present on the nathosoma of the mite, the mouth part, and the body is the ileosoma. So based on this sputum, it may be rectangular, it may have 2 different setal pathways, it may be somewhat rhomboidal in shape. So based on that, they have identified this. So in the southern part of India, Microbiology, this is the major vector that it is abundant, and also this harvests this bacteria a lot. So that is what we got. I got one more shrimp yesterday also. There are so many others, but here I have mentioned some. So this much amount of chiggers and these sticks out of the whole rodent I have collected, around 200 rodents. From that, these are all this much percentage I am able to get. And the predominant we have seen, but the predominance of the ectoparasite form was chiggers. Obviously, they are very tiny when compared with the other ectoparasites of bees and ticks. So they will be huge in number. They can occupy more surface area. They will form this H-scar in the rodent also. Also, the streptococcus. This disease basically. It is a tropical disease and it is mistreated and misdiagnosed by other diseases, mainly the skin disease. So the main characteristic is the presence of a necrotic H-scar in the skin. So it is like a cigarette burn that is happening when this mite bites. So what happens ultimately, probably we get some rashes here, we tend to go to skin doctors or we just apply something. But this pathogen of this Olentia will multiply inside and this may affect other organs, finally multi-organ failure also people are addressing. So such a more fatality rate also. And in Devararam district, we got data from Directorate of Public Health Chennai. We got around 5-year data we got, and it is like 30% streptococcus is increasing this monsoon seasons. Because in this monsoon seasons, these rodents will tend to, they may become lazy and they will be, instead of loitering more, they will themselves in one particular place. This chicken multiplication will rapidly increase. So that might be the reason also. Next. It's just molecular identification. Once I obtain the ectoparasites, I just wanted to identify the pathogen that it is present in the host also. So here the reservoir host of rodent is playing the role. So after sterilizing them, their organs will be isolated and also the blood samples along with the harboring ectoparasites on them. So that there will be no virus. So whatever I get in this pathogen should also be there in this or not is what my interest. So I did real-time PCR after the extraction of DNA. Next slide. So this is the result. Like we had collaboration with Tata Institute for Genetics and Society, TICS Bangalore. So they have designed an assay wherein the clinical samples of streptococcus, what the people are getting, whether the hospital clinicians what they are getting, whether the blood sample has not only Olentia but also leptospirosis, because leptospirosis is also a nature disease that is caused by this rodents. So that's what their interest is. So whether because along the time we found that patients' blood when they screen for this ELISA and all, they got positive for Olentia at the same time they got positive for leptospirosis. they thought there may be co-infection. So that is another strategy where we struggle that we think only one pathogen is present inside our body, but the case scenario is different. We may be infected with more than one pathogen also. So that is, this co-infection is also happening along the side. That is a very big burden also on the other hand. So in order to reduce that, they thought in one assay only we can able to detect both the pathogens present or not. Then the treatment would become very easier and the patient's recovery time also will be reduced. So that was the strategy for them. And they designed the Leptospira and Organised both bacterial detection at the same time. So for that, this real-time assay worked. And it is fortunately in the mite samples we got both the bacteria positive. And we wonder whether this mite is responsible for causing Leptospira or Leptospirosis also. Because this Organised is an intracellular bacteria. So that it just should be there in the mites because to cause the bacteria. That is the other base. But Leptospirosis is biotic bacteria. We have biotic bacteria. Its presence in mite is interesting. We have to delineate it still more. Some more vector competence study has to be done to complete to tell that vector may be mite vector may be the vector for Leptospirosis also. But that has to be a very big another study that we are running. But I got positive in mites for Leptospirosis also. So that is the results. And total proportion of 14.5% of the mites were co-infected with Organised and Leptospira, revealing concerns about overlapping resistance and potential challenges in diagnosis and treatment. Also, approximately 31% of the mites were found to harbor Organised. This shows that the place is endangered was bronchitis. And over half, 54.5% of the mites tested positive for Leptospira species. Next, indicating the findings underscore the dual vector capacity of mites in transmitting both Leptospirosis and Streptococcus pathogenesis. And the statistical analysis with respect to the areas of by determining using the chi-square value, it shows 8.71, it shows significantly statistically significant. So from this, I need to conclude. I have to extend the study to prove whether mites is vector for Leptospirosis on the other hand. It is a quite difficult thing to delineate that, but it has to be done because we should know whether that is a vector that is happening side by side. We have to explore that way. So more focus on the entomological surveillance is happening one side, control measures is happening one side, but the interventions against Streptococcus is still pending. There is no vaccinations also, and the disease is emerging day by day, but awareness is very limited. The thing is, we have to think in a multiple angle that whether we have to stop this mite emergence or mite species diminishing we have to do, or we have to control the pest rodent, or we have to remain hygiene and we have to be like man-vector contact should be stopped. But this cannot be done because in the battlefield man-vector contact will be there. Rodent, even at our homes, we tend to have rodents even when we keep our home clean. Somewhere or the other they enter inside and they make a lot of them. So man-vector contact cannot be, I don't think so, we cannot try that way. But acaricides, mites. So acaricides can be done, miticides can be brought, and rodenticides also can be enhanced so that the target to intervention was practiced to be efficient so that this disease has to be stopped. That is the thing. So targeting humans with vaccination can be done, targeting mice with acaricides, targeting rodents with rodenticides. So in this angle, the question has to be thought and we should enhance programs and other training events on eliminating this disease. NVPDCP also we have to include this disease, one of the 6 diseases that is already designated there. That is all. So this is the picture of my mind taken from fluorescent microscope. Thank you.
Any questions, sir?
You want to ask? Yes. Go, Mike. She want to ask question.
Madam, I want to ask about the
Yes, please.
Borne molecular classification.
Yes.
We have any geneticide. It causes immediate or instant death to the rats without causing hurt to the animals very likely to consume it.
Yes. Actually, DEET, some rodenticides are there. We can keep this, what is that, paste we keep, right? That is also there. DEET and some other chemicals are there, but they are harmful, yet we cannot say that we can only rat eats that. Even if we catch, we keep trap also. Not only rat comes inside, squirrels, some other snakes sometimes comes inside. So we cannot promptly tell that only this should have consumed this particular thing. But rodenticides like DEET is there, that is we effectively used. But in case of paddy fields and all, it is not intervened actually, because that may have some impact on the fields. So it is likely to avoid open cases and keep the waste hygiene and make sure that rodents enter. We should not have contact with them. That is the only case. Yes. Also, for leptospirosis also, it is transmitted from the urine of the infected rat. So our contact should be limited. That is the only easiest way without harming the recipient. Yes, sir. Yes. Yes.
Which is the main course? Might is the main course.
Mite is the main host. Rodent is the reservoir host. That doesn't, this mites present will not harm the rodents. They are just there as vectors.
Only by the biting of mites to the human being that bacterial species will be transferred to the human.
Yes, yes.
So it is an opportunity course.
Yes, yes.
Human
Obviously.
being is an opportunity.
Yes,
Okay. But the main host will be the mites
yes.
for that bacteria. Thank you.
So this Orencia has several strains also. Pathogenium. That is also one of my objective to detect which strain is prevalent in my area.
They are feeling that human being
Not
those who are eating bad.
people are consuming rats also.
That micro in their body.
No, sir, not like that. If we consume rats, I went to some field near Satna, they sell rats. So initially I couldn't find rats in the rat traps. So I thought where rats will be sold so that I can get the rats, buy them and see, observe the presence of chiggers again. I bought that one rat for 200 rupees. From the field, I went directly to the paddy field. They were putting smoke, digging, they have separate persons for that. They were digging, putting smoke and catching, and they catch the rodents and gave me. I got around 6 rats like that initially, but the case is by harming them when they get agitated, this chickens tend to fall off from them. So I couldn't able to get the chickens which were present in the paddy field. Also, because of the smoke they are emitting, these chickens fall off. So from those rodents, I couldn't able to get the chickens. Only from the house rats I could able to get. That is like Rattus volvulus, Rattus musculus. And the thing is,
Question. Tea would more person, time actually. We go for the tea session. We have personal.
Now, please share.
Yes, sir. We have a very good presentation, madam. We have got tea and we have very good discussion there. We go for tea. 5 minutes, we have to start a new session. For that session, Dr. Ramanathapuram and Kodalingam. Dr. Kodalingam will be the co-chairperson and Muthu Pandey, scientist, GR will be the chairperson. we'll adjourn party now. Before that, we would like to honor Dr. Nasaruddin Nasir for giving an invited talk. Shivech, Jayaraj. Shivech sir will hand over the memento to Dr. Sir, ಎಲ್ಲ ಫೋಟೋ you give it the content. Subscribe, come in Maharashtra, he will give it the content. Very useful. You have said it in the program. That's why you will be part of the
Sir, அவர் தலை சொல்லுவாங்க தலை ப்ரோ ப்ரோ ப்ரோ
single
நீ போய் அடியா அப்படி இந்த பக்கம் தள்ளி வாங்க சென்ட்ரும் கொஞ்சம் இந்த மாதிரி கேமரா மட்டும் தெரியாது ஓடிட்டா இருக்கு கிளாஸ் இறக்கி ஒரு இது 45 வச்சது சார் சார்
plasmodium species, which is transmitted by Anopheles mosquito species. And India has targeted to eliminate malaria by 2030 and also global target to eliminate malaria. But the population concluded that in factor to eradicate the disease. And in this study, to understand the population genetic structure and diversity of this data is critical for designing effective control strategies and predicting their response to environmental changes and improvements. And this study focused on microbiology CYG marker to conduct the evolutionary release of the different Anopheles mosquito species and population structure and genetic diversity to gain insight of into the evolutionary relationship and gene across the geographical regions. And our studies are focused on northern part of Tamil Nadu, India, which includes Syed, Krishnagiri and Vellore districts, which is mainly focused on agriculture and playing lands and forestry and rural and urban areas. We conducted mosquito sample collection during July to September 2025. And this is the methodology slide. Collection and what is the process we did to end the study, DNA collection and PCR analysis and amplification and standard sequencing and what is the software we used for analysis, biotech and megawatt with 100,000 bootstrap and we did approtechnic biology. And the results, what is the particular features we collected from that area? Anopheles annulare, Anopheles pallidus, Anopheles obscurus, Anopheles vagus, Anopheles gemti, and Anopheles castellitis. These are the mosquitoes represented on there. And what are the total of mosquitoes we collected? 273. And this is the lineage of the Anopheles mosquito species based on microbiology with 1,000 bootstrap in mega. We use in neighbor joining method. And the species we collected Anopheles vagus and gemisi and Anopheles castellitis Anopheles. These are the two, which is correlated, which is it shows the similarity with South Asian species. And this is annularis complex. We collected two annularis complex. One is Anopheles annularis and Anopheles castellitis. And this is the intrinsic and intraspecific distance of the species. Anopheles annularis and Anopheles castellitis and Anopheles gemisi and Anopheles obscurus and Anopheles vagus. The highest intraspecific distance, which is the most similar to one species to another, it represents in 0.19. The Anopheles castellitis, it shows the highest intraspecific distance value, which is more similar. And the low interspecies distance value, it shows in Anopheles pallidus to Anopheles annularis because both are in same context. So 0.087 is the lowest interspecies distance value. And this is the haplotype network of Anopheles annularis based on the medium joining method using CY sequence in proper sort. There are 24 haplotypes presented throughout the globe. What are the sequence present in the interior? We collected and did the each individual circle represents the haplotype. This haplotype is called due to the genetic variations in between the same species. So we include the species from India, Thailand, Sri Lanka, Bhutan, Pakistan, Myanmar, and Indonesia. There is no more haplogroups within this, but there is a lot of haplotypes. And haplotype network for Anopheles obscurus based on period. And we include the sequence from India, UK, Pakistan, Sri Lanka, Thailand, Europe, South Asia, and Bangladesh. And there are 50 haplotypes are present in this Anopheles obscurus, which is the dominant species in Tamil Nadu. And it exists in A form and B form, C, D.
And the Anonymous Subcutaneous A is the most popular in regions or regions in the inland of the Tamil Nadu. In coastal areas, we see the Anonymous Subcutaneous B group. Here, our sequence matches the Anonymous Subcutaneous A group, and other we mentioned the Anonymous Subcutaneous B. And this is another Vega, which is this is also the dominant species in our region, and it exists in two haplogroups, which is it contains more than one haplotype, means haplogroups. So it within we include the species India, Myanmar, Sri Lanka, Thailand, China, Vietnam, Bangladesh. We include the southeastern countries, and it exists the genetic diversity within the haplotype 15 and 17 genetically diverse and to form a few haplogroups. Next. And this is the another Spiders haplotype, and there is only species from India and Sri Lanka there. And it exists 14 haplotypes, which is closely associated with the Sri Lankan species. Haplotype network for Anopheles jersey. There is 19 haplotypes. Include India, Thailand, Myanmar, Sri Lanka, China, Vietnam, Cambodia. And this is the species which is minimum haplotypes. When compared to the total number of sequence, it shows the haplotypes. And the haplotype 17 and haplotype 16 comes from haplotype 16 is from China and haplotype 17 from Sri Lanka. There is huge mutations occurs in between. Every single strike between this each haplotype, it is represented mutation. And haplotype network of Anopheles tessellatus. It includes India, Myanmar, Thailand, Belgium, Sri Lanka, Japan, China, Vietnam, USA, Indonesia, Malaysia, Singapore. This species exists 4 haplotypes. In Indonesia, there is a separate haplogroup which is diverse from the other haplogroups. And in India, there is one haplogroup which represents in the haplotype 73. This is the India species, and this is the first molecular evidence of Anopheles tessellatus from this study. And next, and this is the overall result, which represents number of haplotypes and polymorphic sites, number of mutations and singleton variable sites, parsimony informative sites, and nucleotide diversity, haplotype diversity, and gene flow statistics, and ATGC content of the DNA sequence, and Tajma's gene significance. And here, the highest haplotype diversity occurs in the species of Anopheles vapors, 0.976, and the nucleotide diversity is 0.0335, and the lowest haplotype diversity occurs in Anopheles pallidus. There is 0.735, and the nucleotide diversity is 0.007, which is low. And the significance between this genetic diversity present in the p value, p represents the significant value, the star is significant, and others are not significant. And Fujiti's test, FST statistics, the minus value represents the low gene flow present, and the highest value represents the high gene flow present across the population. Here, the lowest values occur in Anopheles tessellatus, minus 36.76, and the highest gene flow occurs in Anopheles gems. When compared to the other species, and the studies, the overall studies represents the high genetic diversity which exhibits means negative, means rare allele spread within the populations. And the pattern consists with recent population expansions or selective sweeps. And the strong values occurs in Anopheles vagus, Anopheles tessellatus and Anopheles subpictus. It reflects the high genetic variability or recent demographic events related to the vector. And I thank to our funding agency ICMR and DBT. Any questions?
Nice presentation. Where did you identify the species?
We identified the species in Hosur IVCZ, Institute of Vector Control and Zoonosis.
What about the malarial vector, Anopheles typhi?
Yes, here the Anopheles hanularis is the secondary malarial vector. And recently Anopheles subpictus and vagus also reported as vector in some published paper. And they are infected with Plasmodium sporocyte protein. So that's why. Three species are mainly focused. Anopheles annularis, Anopheles subpictus and Anopheles vagus. In some of the countries, due to the genetic diversity variation, so that non-vector also converting to a vector. It is unpredictable thing, where it is, how it is. So that's why we focus the genetic diversity. It is essential for insecticide resistance also.
Okay, you compared with India data with Ammal with
Yes, sir. Yes, sir.
South Asians.
Yes.
Which country mosquito population is more diversity? Based on your study.
Usually depend on our study. Thailand is the main country to have more diverse, and then India.
India.
Yes.
What will be the reason for?
Because there is climatic difference and geographical region, which is in one place there is a high altitude, in another place there is low altitude, and so seasonal and variations.
The high altitude, low altitude, seasonal variations may increase the variations.
Yes.
But you said there is less variations in Indian populations.
No, sir. In India also have high diversity.
Okay.
So we know we did
Okay,
not.
how is this related to malaria
Yes.
in that particular region?
In Malaria, in historical events, there is a high incidence in also incident in Krishnagiri district and Dharmapuri district. Also reported.
You have data.
Yes, it is. Yeah.
So among the anomalies, mosquito is causing the anomalies, female anomalies, mosquito is causing malaria. But there are different types of species out there. And in what way that we have to identify the symptoms caused by a particular anomaly species. So anomalies is causing that a particular malaria is spread by the biting of that anomaly mosquito. And there are the microorganisms that are causing the Plasmodium, the protozoal microorganism is causing that particular disease. But we can differentiate that particular species of anomalies with bite that patient by means of the symptoms of that bee. Have you any idea about that? Because that sometimes these symptoms may be 48 hours. That malaria will be caused. Sometimes the diversity, what is the, because the anomalies female mosquito is causing the disease, but there are different types of Plasmodium species are there. Depending upon the different type of Plasmodium, and we can understand the symptom. When a doctor will be consult the malarial fever, that symptoms will be easily identified, and he will give the information about the which species of Plasmodium has been transferred from that anomalous mosquito. Can you able to identify that? Only sometimes the fever will be appearing after the biting of 48 hours. Sometimes the fever will not appear 48 hours. But sometimes that fever will be appearing after 72 hours. So with these differences, one can easily we easily understand that the malarial fever has been caused by the biting of Plasmodium vivax, that vivax malaria. And another important one is the fever will be caused by the Plasmodium malariae. Another difference is that malarial fever has been caused by the Plasmodium bovae. So that can be easily identified and the treatment will also be given in a different manner. Can we any idea?
We focused on vector level, sir. Parasitic.
Diversity of the mosquitoes that we have studied.
Yes, sir.
And that is causing that malaria fever. You are aware of any idea?
Actually, there is a lot of studies going on to identify the parasite in vector level.
Narrated only the diversified malarial the Anopheles mosquito. Which mosquito is causing that particular 48 hours malaria by the biting of that malaria mosquito. There are different types of mosquitoes out there. But are you able to
The question is, reporting from where you get data for the international population?
Sir, it's
Data is provided by CMR or you can get it?
no, sir. We collected the data from public database, NCBL database, and then National Institute of Health.
You mentioned the international varieties of mosquitoes.
That was a nice presentation. Madam, you go and sit next to Dr. Kasina. Call the next person. Man, Gauri.
Good afternoon to all. My name is Gauri. I am a research scholar, Department of Bioinformatics, Ayanappa University, Karachi.
Next slide, please.
We are prostate cancer. Prostate cancer is one of the emergency cancer because it is one of the death-causing cancer. So prostate cancer is one of the uncontrolled growth of abnormal cells in prostate gland. There is
I can take this protein through PDB and I can validate this protein through Ramanathapuram blood. So it is the most favored region is 88.9% and the disallowed region is less than 1%. So this is my result. So I can do single docking. So my docking scores are -4, -5, -5 and another plant. So I can molecular weight bioavailability. What is that observation through the human body and hydrogen by donor, acceptor, log P and the toxicity also I can validate. So and I can do conjugation logging. I can conjugate I can conjugate 13 components. Out of 13 components, this camphorol is hydrophobic, vanillic acid is hydrophilic, and pyridine hydrophobic, vanillic acid is hydrophilic. Show good result through docking. So it can shows 9.85, 9.577, and the bioavailability is also increased, and it also toxicity also same, and it have 3 hydrogen bond regions. So it can merge into hydrophilic components. By taking hydrophilic components, it will be easily digested into the human body and it have less side effect. So it is also run through molecular dynamic stimulation. I can find 2 different components through RMS stimulation, and RMS fluctuation also done in 500 nanosecond. It can stable throughout that experiment. So further in vitro studies was ongoing.
Very good bold and voice modulation is very good. But my doubt, you are pretty much concentrated about hydrophobic, hydrophobic.
Sir, my research in this cancer treatment is cost very high. So we can get medicine through our Indian plants and it also have less side effect. So I can choose our Indian plants and I can take hydrophobic, hydrophilic natural components. Then I can conjugate and use it for prostate cancer treatment.
Your point, which will be the good?
Sir, both will be good. By for taking hydrophobic component, the side effects are very high. So if we conjugate it means the side effect will be reduced, the cost is also reduced, and it is useful for all people. So this is my.
You mentioned this curcumin as a hydrophilic or
Curcumin is a hydrophobic component.
But it will be digested in our body, no?
So it will be digested like 60 to 70% only. That many are in the body itself only. So it will cause a very severe side effect future. It will cause future because it is digested in our body only. So it will cause some side effect. So only we can conjugate it, the side effect will be reduced, and also bioavailability also increases. So bioavailability increases means we can cure the prostate cancer treatment to the next level.
Okay, what is the conventional chemical-based treatment used for prostate cancer?
Sir, chemical-based
Not only plant. Conventionally, they use chemotherapy. You know which compounds they are used. You also mentioned what are the available medications, what will be the side effects, so that you focused for phytochemicals. Justifications. Thanks.
In traditional medicine, we have so many plant products are there. What could be the reason for selecting the particular plants to identify?
Sir, not particular 2 plants. My work is mainly about conjugation. Conjugation of any 2 Indian plants and make one new component and use this component for this treatment, prostate cancer treatments.
Can you tell me any 2 side effects of that?
Sir?
Any 2 side effects? You mentioned side effects, no?
Yes, sir.
Any 2 side effects?
Sir, it will cause like kidney will damage, liver will damage. The intake of so many hydrophobic components, it will mostly 60 to 70% are excreted, but remaining are in our human body. So it will like cause liver damage, kidney damage.
So that
Is there any evidence for it?
yes, sir, is there paper evidence is also.
Good afternoon to all present here. I am CM Organised Research Scholar from Wool's College, Vellore. My presentation topic is phytochemistry, insecticidal and phytoremediation properties of Sapinus mucorrhizae. Next slide. This is the introduction of this Sapinus mucorrhizae. It is a deciduous medicinal tree that is distributed in tropical and subtropical regions of Asia. It is commonly known as Rita in Bengali in English. Sorry. It is commonly known as Rita in Bengali in India. And then the vernacular name of the Sapinus mucorrhizae fruit is soapberry, Chinese soapberry and soapnut because of the use of the pulp in soap making. The fruit is pericarp and the remaining is seed. There are 56% of the fruit is pericarp. Then the fruit becomes reddish brown. Next. In my topic, there are three divisions are there. First one is phytochemistry of Sapinus mucorrhizae. There are various compounds have been isolated from the different parts of Sapinus mucorrhizae. From that, the fruit has more amount of saponins, that is 11.5% of saponins, and it contains 10% of sugars. Apart from the saponins, it also contains three components such as fatty acids, flavonoids, oligosaccharides. Some of the uses of active compounds that are gained from the Sapinus mucorrhizae that I have collected from the literature papers, that is oleic acid. It shows antifungal, anti-inflammatory and antibacterial properties, and then the tetrotetonic acid that also contains anti-cancer and antioxidant properties. Then the pentadeconic acid contains anti-cancer activity. Then methodology. Collection of the plant parts. The Sapinus mucorrhizae plant leaf and bark were collected and gathered from my college, Wool's College from Vellore district. Then the surface sterilization is done by using 0.01% of mercury chloride. Then 70% ethyl alcohol were used for 1 minute. Then aseptic blotting paper was used to dry the surface sterilized leaves. And then the barks of the Sapinus mucorrhizae. Then it can be sterilized and double distilled water can be used for cleaning purpose. Then preparation of the extract. The sterilized leaves and barks were shade dried for 10 to 15 days without exposure to the sunlight. Then it can be powdered by using the electrical grinder for leaf and bark extraction. Then it can be dissolved in the 50 ml of methanol and stirred vigorously at maximum speed for 3 hours at 60 to 70 degrees Celsius. Then it can be filtrated by using for methanol evaporation on a transparent petri plate after the setup was left alone for a full day. Then the extract was ready to subject to the GCMS analysis. The GCMS analysis, that is gas chromatography and mass spectrometry analysis, this was specially done in the fish lab in MKG College, Warnimbaadi. Next slide. It was reported that the phytochemicals are the bark extract of Sapinus mucorrhizae by GCMS analysis. In this extract, there are 11 compounds or active compounds are extracted from the bark extract. Sorry, there are 11 compounds are derived from the bark extract of Sapinus mucorrhizae by GCMS analysis. These are the compound name, that retention time and molecular formula or molecular weight of these 11 components from bark extract. Next slide. These are the chemical structures of these phytochemicals that are derived from the Sapinus mucorrhizae. And then the phytochemicals derived from the leaf extracts of the Sapinus mucorrhizae by GCMS analysis. Apart from that bark extract, presentation. It contains there are 16 compounds that are derived from the leaf extract of the Saccharomyces mucorrhizae by GCMS analysis. These are the compound name, retention time, and molecular formula and molecular weight of these phytochemicals. Then next, these are the chemical structures of the phytochemicals that are derived from the leaf extract of the Saccharomyces mucorrhizae. Next. Then we are going to our second division of this presentation topic, that is insecticidal activity of Saccharomyces mucorrhizae. This Saccharomyces mucorrhizae also contains insecticidal activity. For example, the Saccharomyces mucorrhizae contains the insecticidal activity against the cotton leafworm, that is Podoptera librolis, caterpillars, and the P. Acethrosiphon poison, and also it contains insecticidal activity against Cytophilus oryza, that is rice weevil, and Pediculus humanus, that is headlice. And also it contains it acts as a biocontrol agent of the insect Graphiolata mollusca, and also it kills the larvae and the adults of the Graphiolata mollusca. The saponins also can act as a biocontrol agent for insects, and also it is act as a potential insecticide, and it deters the feeding, growth, and egg laying process of the different crop feeding insects. These are the insecticidal properties of Saccharomyces mucorrhizae that I have collected from the literature papers for the last 10 years. These are the different scientists that were discovered, many researchers on this insecticidal property of Saccharomyces mucorrhizae. Next slide. Then this is my last division of this topic, that is bioremediation activity of Saccharomyces mucorrhizae. The compounds and the enzymes found in the Saccharomyces mucorrhizae tree could offer an alternate option for the removal of harmful chemicals from the environment. For example, it is very useful to remove the various pollutants such as polycyclic aromatic hydrocarbons, dyes, phenolic compounds, and heavy metals like chromium, lead, arsenic, copper, and zinc. We are going to concentrate in the remediation of heavy metals. The sources of pollutants are mining, use of herbicides, pesticides in farming, burning, fossil fuels, and effluents from textiles, paints, and metal industries. These heavy metals are carcinogenic, mutagenic, and can cause lethal damage to vital organs like liver, kidney, heart, and spleen, and etc. In this regard, Saccharomyces mucorrhizae could be an important plant that can help in remediation of these heavy metals from the environment. Several studies have shown the role of Saccharomyces mucorrhizae in the removal of the different heavy metals like arsenic, chromium, lead, and zinc, and etc. This table column shows that several scientists have discovered bioremediation properties of Saccharomyces mucorrhizae for the last 10 years that I have collected from the literature papers. This is my plan of work I am going to do in my upcoming research work. That is, I want to reduce the usage of harmful and chemical pesticides which are used in various agricultural fields. So I choose the agricultural fields present in my native place, Gudiyatham. In Gudiyatham, various types of crops are cultivated. Some of them are paddy, groundnut, sugarcane, various fruits like mango, banana, and then vegetables like brinjal, tomato, and these are the colored lettuce that are brown panthoper, yellow stem borer, leaf hopper, whitefly, scale insects, and fruit fly, banana weevil, aphids, and stem borer. These are the pests that affected crops in my native place. So in this agricultural field, the chemical pesticides are used. Some of them are I mentioned here, that is peptide, pidoclaprid, and chlorpyrifos, and etc. In spite of using these chemical pesticides, we can use the pesticides derived from the Saccharomyces mucorrhizae because it is eco-friendly. Next. Then I am going to focus about the bioremediation property of the Saccharomyces mucorrhizae. For example, I taken in the Ramanathapuram district, chromium contamination is identified in many areas due to leather canaries as well as the chromium canaries. In order to remove the heavy metal chromium from the contaminated soil, the active compound separated from the Sapinus mucoraceae may use it as a facilitator. So I decided to eradicate the chromium from those areas by using this active compounds of the Sapinus mucoraceae. So in my upcoming research work, I am going to get the solution for these problems by getting these insecticidal properties and also the bioremediation properties of the Sapinus mucoraceae in my upcoming research work. Thank you so much.
சார் ஒரு கொஸ்டின் நான் கேட்டதே இல்லை. இது தமிழ் நேம் நம்ம சாப்பிட்டஸ் இண்டெக்ஸ்
சார்
சாப்பிட்டஸ் இண்டெக்ஸ் தமிழ் நேம் சோப்புக்காய். நீங்கள் ஒரு நான் கியூரியாசிட்டி தான், இந்த கொஸ்டின் வந்து கியூரியாசிட்டி தான். இதில் உங்களோட ஃபைண்டிங் என்ன சொல்ல வரீங்க? இன்செக்டிசைட் ப்ராப்பர்ட்டி இருக்குதுன்றீங்க. இது நான் ஒரு புக்கு படிச்சு கிட்டத்தட்ட ஒரு 20 years முன்னாடி பூவுலகின் மைந்தர்கள், பூவுலகின் நண்பர்கள் ஒரு புக்கு. அதில் வந்து நம்ம பாண்டிச்சேரி யூனிவர்சிட்டியில் ஒரு ப்ரொஃபஸர் வந்து ரொம்ப பெரிய ஆள், ரிட்டையர் ஆகிட்டாரு. He has written one article where in the Africa the tribal people used to put this and wash the clothes. There will be, you know, the large giant snakes will be there in the entire. That giant snakes are the vectors of so many diseases that we all know. I don't want to go into the background. So he was on the way, that one scientist was on the way on the riverside, and he saw one fine morning, he saw all the snakes were dead. All the snakes were dead. And then he found out that what is the reason, and he found out that this Sapinus mucoraceae is the reason why where the tribal people are washing their clothes. இவ்வளோ செத்து போய் 25 வருஷத்துக்கு முன்னாடி சொல்லிட்டாங்க. உன்னோட வாட் இஸ் யுவர் நியூ ஃபைண்டிங் இன் தட்? தட் இஸ் வாட் மை கொஸ்டின் இஸ். சோப்பு காய் எல்லாருமே தெரியும், சோப்பு காய் நான் கூட தலைக்கு போட்டு போயிடுறேன். அதில் கொஞ்சம் நாளைக்கு முன்னாடி வந்து சிகர்காய், யாரோ ஒரு மீரா சிகர்காய், அது எல்லாத்துக்கும் அதான் இருக்குது. எல்லாம் ஆன்டிசைடல். என்ன இதில் உங்களோட இதை எப்படி வந்து ரிவ்யூ ஆஃப் லிட்ரேச்சரில் எப்படி நீங்கள் கேப்பை ஃபைண்ட் பண்ணிட்டு ஹவ் யூ ஃபவுண்ட் அவுட் கேப் எல்லாம் இருக்குது, மொத்தம் இருக்குது. இல்லை சார் இந்த பூச்சியில் அவன் சொல்லலை, அதான் ரிசர்ச். இந்த நண்டில் சொல்லலைன்னா அடுத்த நண்டில் சொல்லலை, இந்த கொசுவில் சொல்லலைன்னா அடுத்த கொசுவில் சொல்லலை, அதான் ரிசர்ச் பண்ணுறோம். நீங்கள் ஹவு ஆர் கோயிங் டு ஜஸ்டிஃபை யுவர் ஒர்க்? அவ்வளோதான் போதும் சார்.
Sir, in my college I found this plant. It is very different from other plant species. So I decided in my PG project work.
Already literature based bioinsecticide. Chromium separation in the active component. Chromium.
சரி,
And then I have a question. Why don't you concentrate on the fruit of that particular plant? You are concentrating only on the leaves. The fruit is more essential than the leaf.
The fruit only contains more amount of saponins, which can act as surfactant and also soap making. In my upcoming research, I can also use that soap.
Not only fruit, the bark, the root, and everything.
Then in future we can hope that your findings will make a biopesticide.
Yes, sir.
Isn't it?
Yes,
We are all using the chemical pesticides.
Yes, sir.
The chemical pesticides are normally they are killing the pests, but even though they are causing so many damages to the environment.
Yes, sir.
Isn't it? But if we use the alternative biopesticide, and that will be highly used, it will not pollute the environment. It will only kill the target organism.
Yes, sir.
And the plant biopesticide is the safest one in order to kill that pest as well as to ignite the environment. That is, damaging the environment is very serious. Because without knowing, these people are, many countries are using chemical pesticides. Thereby they are causing pollution in other ways. Because it is chemical, only we are killing the target organism, but at the same time the residues of the chemicals contained will spoil the environment. But if you prove these sorts of biopesticides, isn't it, extracted from that particular plant, if it is used as alternative for chemical pesticide, that is highly useful for protecting our environment. Very good, thank you.
Thank you.
So is it convenient here? Is there heavy metals? You have mentioned that it is a textile industry. Textile effluents. So we open the literature. So in the textile effluents, that chloralkali plant is there. So the main effluent contains
because of the tanning in
The textile industry he told, that's why I'm asking.
all the other options for this. But he is specific on bone.
Sir,
Okay,
Sir, one question. You said you are soaking with 50 ml of methanol. What kind of percentage of methanol you are using for preparation method? Percentage of methanol. It doesn't affect the plant to the ingredients and the banana.
You are presenting. Very good, presenting. Okay. No, you can take 10 minutes also you can take.
Lethal rate.
I thought it is over.
Insecticide you are using. What kind
Okay, I
of
thought it is
lethal
over.
rate? It's a biocontrol agent.
simple
So also it
presentation,
is
no
Okay, okay.
And I have conducted from declaration backers. Going forward.
Okay, Vapani, Budi Pamute. ಇಲ್ಲ ಅವರು ಪಂಡ್ರಾರೆ ಕಮಾನ್ ಸರ್ ಸಾಗರ್ ಓಕೆ
Dr. C.P. Sagar.
Thank you so much. Good afternoon, all of you, respected sir and ma'am, chairperson and co-chairperson. My name is Dr. T.P. Sagar from Satna, State MP. I am working from Rural PG College, MP State. This is my presentation in human sustainability and Vector-Borne disease control. Introduction. Vector-Borne disease such as malaria, dengue and chikungunya contribute to the C10 human health and sustainability development. They study examine the link between human sustainability and vector-borne disease control, focusing two parameters: intervention strategies and environmental factors, mixed method and the approach, quantitative data and the literature-based quantitative aids. Finding revealed that long-term disease control demands ecological awareness and environmental participation and innovation. Next. Introduction Vector-Borne disease. Commonly Vector-Borne disease account of the over 7 lakh deaths in the globally per year WHO 2024. They hinder the productivity, education and the economic sustainability, directly impacting human sustainability. Traditional control methods, insecticide force the dominating return due to the resistance and the ecological dispersion. They integrated vector management approach emphasizes rational, eco-friendly strategies combining the multiple interpretations. One has remarkable integrated the human, the animals and environmental quality of the sustainability Vector-Borne control. Literature singing part on the one integration strategies proving the effective in the malaria endemic resilience up to the 85% reduced in the malaria residence ETR 2021 distribution of position in the DRC the 41% of the decline in the child mortality. Belgium mosquito intervention introduced in the Indonesia 77% the reduced in the dengue residing and the 86% of the fever hospitalization. Integrated program Bacteria mosquito intervention in the Indonesia 77% the reduced in the Heavy rainfall 200 mm weekly, larva survival, outproduction, reduction. Coordination of the urban growth. Next, discussion and the implication. Integrated control along public health, agriculture and environmental sectors, community participation, finds the ownership of the ensure proper net use of the climatic resilience in the relative outbreak. Next conclusion. Integrated control. Next. May enhance human sustainability depend on the balancing ecosystem and strong health system. The combination explain one health and framework of the across all thermal programs, innovation in the eco-friendly, innovation, biological control, genetic methods, organized climatic smart of the urban planning to the minimize mosquito habitats, eco-arrange in the local enforcement of the thought, education and the participation. Handling of the sustainable vector control is not a event, it's on the evolution partnership in the between of the Vector-Borne series. Yes. Thank you so much. I am prepared sometimes. Given the
So you presented a holistic approach how we can manage the vector control, isn't it?
Yes.
In your viewpoint, the Indian government is doing good or yet to improve the vector control strategies? What in your viewpoint?
Yes, only two matters. Okay, improve this matter. Two matter parameters. Not one India and IMP. Only other words of the some matter included here. Yes, yes. Thank you so much.
கொஞ்சம் கஷ்டப்பட்டு அதை தூக்கிட்டு இப்போ பண்ணி க்ளோஸ் பண்ணுங்க.
Nice presentation. In this presentation, everyone could notice that particular point, environment and also Vector-Borne disease. Most probably in last presentation, the eco-balance is very essential for human health. I think so we need to concentrate on public health the same way we need to concentrate on the environmental protection. Thank you.
Thank you, Dr. Muthupandi. I take the honor, pleasure and pride in appreciating and giving a memento to Dr. Muthupandi as a chairperson. Thank you, sir. Wonderful session you held there. Thank you. I request now our best friend, Dr. Kudalingam, to associate with him and to chairing a session. Thank you, sir. And one more information, sir. So far, நான் போட்ட தமிழையாவது புரியுதான்னு நான் கேட்குறேன். குரூப்ல யாரெல்லாம் நாளைக்கு ராமேஸ்வரம் வரீங்கன்னு கேட்டேன். யாரும் ஆன்சர் பண்ணல. எனக்கு இல்லை, நான் காரில் வந்துருக்கிறேன். 3 தடவை ராமேஸ்வரம் போயிட்டேன். நான் போயிட்டு அதெல்லாம் நான் பாட்டு காரில் போயிட்டேன். யாரும் வரீங்கன்னா தான் பஸ் கேட்குது. 40 சீட்டர் பஸ். அதே மாதிரி சாப்பாடு எத்தனை பேர் வந்து யாரும் சொல்ல மாட்டீங்கன்னா நான் இன்னும் நைட்டு சாப்பாடு கிடையாது. நான் எப்பவுமே ரூம்ல வச்சுருவாங்க. யாருமே ஆன்சர் பண்ண மாட்டேன் குரூப்ல. பட் அதான் I know, then only I will come to know the number, then only I will come to know, then only I can ask the bus. Now, I call upon Dr. displayed on the online. You see the name and post the questions in the group itself. Anybody, suppose Ramanathapuram complete that also. Is coming here, and we are waiting for her to receive the memento. That's why we are waiting here. After this, we will be adjourning for lunch, and we will be assembling here by 2 o'clock, 2 to 2:30, concluding session. We are waiting. For you only we are waiting. Sir, there are 4 posters presented. Now I have put it in the group. You want to tell something?
What are the questions?
Yeah.
What are the questions?
Swati Yadav, Janvi and P. Pawar. Three people. The title is not visible.
Swati Yadav, he hearing. Swati Yadav, let me know how many viral blood.
Sir, actually this is not live, sir. This is the YouTube. People will post the questions in the YouTube.
Yes.
Yeah, they will post the questions. Yeah, you cannot ask her. They will not be listening. They will not reply.
But she will ask.
That everybody will post in the. You have to post in your mobile. Sir, then. Sir. Very presentations are over. This poster presentation, they have posted it in the group yesterday. Just for the mark of respect, that I would like to involve Dr. Arun Kumar. That's why I put him as a chairperson. This is a live link in YouTube. They will see, but they cannot respond. So that is a problem now. Anybody who has got the questions can post the questions in the group. Later answer, later not answer. But for our information, we are posting it. Because of the interest of the students, we are posting it. So that they also will have a feeling of presented. Now I call upon yesterday's poster presentation. Offline was judged by Dr. Senthil Kumar on mark of respect. On mark of respect, we would like to honor the chairperson memento and I request Dr. Senthil Kumar to receive the memento from
Sir, send it. Sir, excuse me.
Now I call upon Dr. Arun Kumar for having chaired the poster presentation session to receive the memento for a chairperson memento. Assistance by only a single person, single woman army by Jagakshmi Krishnan. The entire session yesterday, we were all enjoying in the temple, but she and her students were waiting here till 7 o'clock.
Oh.
They have completed till 7 o'clock evening. She was in the Wisdom department studio and she completed all the posters.
Oh.
See, there may be some leftover offline, but none of the papers have been left. But all the participants, online participants, were very happy, and it is because of Dr. Jayalakshmi Krishnan. We would like to honor, and we are most honored, madam, by your wonderful work, and I call upon to receive your memento. And we are missing Dr. Srivatsava, and he being here, he will be very happy. Thank you, madam. Thank you, wonderful.
To see their efficiency.
Very good, very good, very good. We'll all adjourn for lunch, please. Join us for lunch, and we should be back by sharp 2 o'clock. If you want to go around Ramanathapuram, you'll be back here by 2 o'clock. Or else we'll be here till 5 o'clock. You choose the best answer. Come on, let's all have a nice talk.
Sorry, வச்சுக்கூடா
சார் செல்வம் பாருங்க சார்.
நீ இப்படின்னு எடு
நம்ம ஸ்டார்ட் பண்ணிக்கலாம் விடுங்க. நான் செல்வம் பாருங்க.
லைவ் இந்த வீடியோ மாற்றிக்கு 2000 செஞ்சது அவசியம் இங்கேயான வச்சு டக்குன்னு நீ இவ்வளோ போதும் அதை அங்கே வச்சு எடுக்க மாட்டியா? ஏன்னா உள்ளே வரதுங்கிற இங்கே வச்சியா கேமரா இங்கே வச்சியா? ஏன் வை வச்சு நான்
Good afternoon, honorable present here. Sir, we are now time. See, we are running short of time, but we will have a separate discussion after tea. Now, this is the concluding session. I request the most respected Chief Vector-Borne Officer, former Chief Vector-Borne Officer of the Great Chennai Corporation, Dr. Dhanraj, to follow the dais. Sir, welcome. Now, I call upon the Principal of Syed College to adopt the dais. I request Dr. Syed to come around the dais. The guest of honor, Dr. Jayanti. Dr. P.K. Srivastava, please sir. Organizing Secretary, Dr. Rajit. Shivesh, come. Sir, please be seated, sir. Now I give the podium to national anthem. National anthem. One more chair. Chair. Dr. Salupar, no. Thank you there. Now I
Good afternoon. Now I request the principal to give the welcome address.
Sir, yeah.
Good afternoon, Honorable Speaker. It gives me immense pleasure and pride to extend a very warm welcome to each one of you to the valedictory function of the 3-day international conference on life science for human sustainability with a special emphasis on control of vector-borne disease. During the past 3 days, this conference has served as a vibrant platform for researchers, scholars and academicians from different parts of our country and abroad to exchange ideas and present their findings in the field of life science. The discussion and the deliberation have truly deepened our understanding of human sustainability and the control of vector-borne disease. Really, I feel very happy to welcome respected Dr. Syed BC, General Secretary, the Society of Life Science, Satna, Madhya Pradesh, respected Dr. P.K. Srivastava, Director, the Absolute Human Hair Foundation, former Joint Director, Ministry of Health and Family Welfare, New Delhi, respected Dr. S. Selvakumar, Chief Vector Control Officer, Greater Chennai Corporation, Chennai. I extend a cordial welcome to our guest of honor, Dr. J. Jayanti, Dean of Science and Head Department of Advanced Zoology and Biotechnology, Purnana College, Chennai. With due respect, I would like to welcome our respected Dr. Fatima Sanas Farooq, member of Syed Ammal Trust. We are very grateful for your presence, and I also extend a warm welcome to our Chief Guest, Dr. P. Dhanraj, former Chief Vector Control Officer, Greater Chennai Corporation, Chennai, who has kindly accepted our invitation to deliver the valedictory address. I extend my special welcome to all the faculty members, research scholars, and the students delegates who have actively participated and enriched the session through their valuable papers and discussion. My sincere welcome also goes to Organizing Secretary and Head of the PG Department of Microbiology and our Senior Most Professor Dr. Ramanathapuram, who along with his dedicated team has played a major role in making this international conference a grand success. As part of this valedictory session, we also have feedback from the participants, and I request all the delegates to kindly share their valuable impression and recommendation about this 3-day academic event. We are also proud to recognize and honor those who have been selected for the Best Paper Presentation Award. Your contributions have truly added the value of this international conference. In addition, we are privileged to witness the presentation of the Lifetime Achievement Award conferred by the Society of Life Science, Satna, Madhya Pradesh, and the Absolute Human Care Foundation, New Delhi, to distinguished personalities who have made remarkable contributions in the field of life science and public health. On behalf of this Ammal Trust, the faculty members and the students of our college, I once again extend a warm and a cordial welcome to all the dignitaries, guests and participants present here on this special occasion. Let us carry forward this spirit of collaboration, innovation and academic inquiry that this conference has inspired as we continue to work together for a healthier and more sustainable future. Thank you, one and all present here. Thank you so much.
The next session. Now I request Nandit sir to give the memento and principal sir to give the shot. To honor the chief guest on the shot, I request Dr. Dhanraj and I request Dr. J. Jayanti ma'am.
Excuse sir. Sorry. Sir, inge parunga sir.
I request Dr. J. to receive the honors from the principal, the shawl, and member I request Dr. J. Jayanti, Head of the Department of Advanced Zoology and Biotechnology, Dean Sciences, to give away the address for the gathering.
Thank you very much, Dr. Vector-Borne, for wonderful comparing that you have did and for bearing with us for the past so many days. We kept changing the schedule and we kept making a lot of changes here. You were there, adjusting to whatever we wanted, and that was wonderful comparing. Before I start, I would like you all to give an applause for the compiler, Dr. Vector-Borne, and encourage such talent which is present among us. Respected chief guests, distinguished delegates, eminent scientists, academicians, research scholars and dear participants, a very good afternoon to all of you. It gives me immense pleasure and deep satisfaction to stand before this august gathering and deliver the concluding remarks of this prestigious scientific meet on life sciences for human sustainability with special emphasis on control of vector-borne diseases. Before I talk about my remarks, I would like to talk about this particular conference. Already, I think Srivatsa sir and Syed sir have already told regarding how this entire thing started, this entire concept of the conference started. I think as far as I remember, we have all had gone for a conference now, and during the deliberations and after the post-deliberations, I think, we were discussing, and in 2012 we had conducted one such thing with the Zoological Society of India in our college, and subsequently when Syed sir and B.D. Joshi sir was speaking and Srivatsa sir spoke to sir, and we wanted to have a similar, if at all, a more effective one in South India, but he is the boss of all of us. So it's a good team. You can always vouch with it, because I think when Sir was here, I think 2-3 years back, it was when the Indian Science Congress conducted an event over here. Then the former principal was there, and then Sir would discuss with them, and he would know they are doing it very well. Truly, Sir Balakrishnan Sir, I would like to say that start from only my first time to Syed College. Sir has been there, he has told a lot of things. We have a certain trend to work, and the finish is always very good, and perfection is expected of. So we expect a lot of things. How will you do it is what we were thinking about. But truly from my heart I am saying, sir, right from the hospitality, I am not making this a thanks, vote of thanks or something like that. It's as a participant I'll tell it, that right from the time they received us, you should understand we are part of them, a part of the team. But right from the time they received us, the hospitality, the way it was carried out, the warmth that was given, and every single way it was carried, right from the trustee madam who had come, she absolutely humble person. I have seen a lot of administrators, absolutely humble. She was more welcoming on thought process. She used to please encourage us if you want to do more. That's how she was saying, followed by principals who always received her very, very well, and he made sure that we are comfortable in the campus. And every single time we used to enter, sir, we used to tell, let's go see online session how it is, let's see offline session how it is, then let us see the participant. Only one reply came from them. I think either principal or also Ranjit sir also, only one reply came from them. It will be done. It will be finished. That is the only reply I got it. Mostly in my college I will never accept that thing, because I get scared because whether the person will be able to do it. But I'm telling you, the next day we came and we saw everything was so impeccably arranged that we had but no choice to agree upon. We have found a match. That is what we have found. So it was very, very well organized. Yes, we supported them, but they took it forward on their shoulders, and you definitely would agree upon that it is wonderfully done. Congratulations to Balakrishnan sir for wonderfully leading the team and making a very good international conference. And Ranjit sir, man of very, very few words. Very few words, but I think a man of action. So congratulations to you, sir, and wonderfully doing it. Always we have to support. They usually say success, there is some women behind it or man behind it or something. Always, but here's a man behind or many successes to come and many successes that we have achieved. It's a good team. For many to say it always. No political agenda, but MGR is always a good team, so we have always maintained that rapport, and we have always delivered whatever assignments we have taken up. Thank you very much, and I would also like to thank our team of scholars, which I think mostly I forget to think. Definitely I would thank my team of scholars who are there always to support us. A very big round of applause for them, so please congratulate them. And I would like to mention regarding this entire theme that was taken up, a very surprising theme, but yet a very, very effective theme that was taken, which contributes a lot to social relevance and to the society as such. So I would like to mention few of the gatherings I have taken from here. The human sustainability is about harmonious coexistence with other species and ecological systems that support all life. In this context, vector-borne diseases remain one of the greatest challenges to both human health and global development. Vector-borne diseases continue to claim lives and strain healthcare systems, particularly in tropical and subtropical regions. Technology alone cannot bring sustainability. It must be integrated with awareness, education, and social responsibility. The advances in molecular biology, biotechnology, genomics, and environmental science discussed during this conference remind us that the solutions are within our reach if only we bring together scientific innovation, public health, policy, and community participation. The life sciences community today is moving beyond mere disease control to disease prevention, ecological management, and sustainability-focused interventions. Whether it is the use of biological control agents, genetic modification of vectors, or eco-friendly habitat management, each approach represents a step forward toward a healthier, more sustainable future. The theme of this international conference is both timely and significant. Human sustainability is no longer a theoretical idea, it is a global necessity. As the challenges of climate change, ecological imbalance and emerging diseases intensify, the life sciences assume a pivotal role in ensuring not just survival but sustainable well-being. The insights shared here reaffirm the idea that the future of disease control lies not in isolated efforts but in integrated, sustainable and collaborative approaches. The life sciences today are evolving from a pure analytical discipline into a unifying force that bridges molecular understanding, environmental stewardship and social responsibility. The deliberation during this meet have shed light on innovative and integrated approaches for vector control spanning molecular biology, ecology and public health strategies. We have also discussed climate-linked disease modeling, consistent surveillance pattern and community participation strategies which collectively form the backbone of a sustainable vector management framework. As scientists and researchers, we have a dual responsibility to pursue knowledge with rigor and to apply it with responsibility and compassion. Science separated from ethics or sustainability can never serve humanity in the long run. However, even as we celebrate our scientific progress, we must remember that the ultimate goal of science is to serve humanity. Our discoveries and innovations must reach the most vulnerable sections of society, particularly in rural and resource-limited settings where vector-borne diseases are most rampant. It is here that the true measure of our success lies in the transformation of knowledge into tangible public health outcomes. Before I conclude, I wish to extend my heartfelt appreciation to the organizing committee for the tireless efforts in orchestrating this event so seamlessly. My sincere thanks also goes to the distinguished speakers and participants from across the country for the valuable contribution. Your enthusiasm, dedication, and intellectual generosity have made this international conference truly memorable. Let us depart from this gathering with renewed energy and a shared vision to harness the power of life sciences in creating a healthier, safer, and more sustainable world. Together, let us ensure that our science continues to serve humanity and that our pursuit of knowledge always honors the balance of life. I would like to conclude with the words of a great leader, Mahatma Gandhi. The future depends on what we do in the present. May our present efforts in the life sciences pave the way for a future that is healthier, safer, and more sustainable for generations to come. Once again, I extend my gratitude to everyone who made to this event a grand success. Wishing you all continued excellence in your research and professional endeavors. Thank you, one and all.
Thank you, ma'am, for being here and for the heartfelt talk filled with positivity. It means a lot to all of us. Thank you, ma'am. I welcome Dr. M.G. Ramanathapuram, sir.
Sorry, this is in my agenda. This is an added agenda because I'll not steal more time, sir. It's my proud pleasure. This vector-borne diseases for third international conference was held in Laila College. And that time I met Dr. Srivastava. The organizing secretary is Dr. Raja, assistant professor of the Department of Zoology, Laila College. So on behalf of all of us, I'd like to give a small memento to Dr. Raja. Now I request Dr. Selvam to hand over the memento to Dr. Raja.
Please.
Thank you.
And you all know that the conference was conceived by me, but though this was conceived by me, this was fed and fed by Dr. Selvam. He is the man behind that, silent man behind that. And the management has taken the pride of honoring 11 people. They asked me to give a list of very important people. That time Dr. Selvam was very busy. On behalf of him, I received the memento. Now I request Mrs. Selvam to come to the dais and Dr. Selvam will together receive the memento. Sir, please come. And I request Dr. Dhanraj to hand over the memento to both of them. By YouTube all over India.
Very nice.
Thank you.
Ma'am, thank you, ma'am.
Thank you.
See, everybody was appreciating what our principal said, and he said he was about whatever that. When there is no cooperation anywhere in the host institute, this cannot be come to a realization. I take the pleasure and honor that Greater Chennai Corporation would like to honor the eminent dynamic person, Dr. Balakrishna, for giving us a free hand. Now I request Dr. P.K. Srivastava to honor him with a shot and a memento by Dr. Dhanraj.
சார் இங்கே பாருங்க சார் சார் இங்கே
Again, a man of army, man of action, and a man of silent action is my beloved friend, head of the department of PG department of microbiology, the organizing secretary, Dr. Ranjit Kumar will be felicitated by, he will be garlanded by Shvesh Pratap Singh, and the memento will be handed over by Dr. Dhanraj. Give him a big round of applause.
சார் 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 கத்தியா
Everybody was telling MGR sir, did everything. MGR sir is nothing. Because it is behind me Dr. Jayanti and my team of beloved kids. And I would like to honor my love and affection through the small memento. I request Dr. Dhanraj to hand over the memento to my dear Rebecca Vaishali to receive the memento from the Dr. Dhanraj. Oh. Sir, wonderful work extended by these three kids. And this was the idea conceived by Dr. Sellupar. Sir, Dr. Sellupar only told, sir, these people should be felicitated. And sir, you also stand there. Please. Yes, now I call Ms. Jayashree to receive the memento from the chief guest, Dr. Dhanraj, because you all know that the other person is Dr. Jayashree Krishnan, who entire session of online was supported by her, and then she was Mrs. Jayashree was assisting her throughout the session, and all the typing, everything she only did that. In honor of that, we would like to felicitate her with the memento. And none other than that, very important, once again, my beloved student, once PhD research scholar, Mrs. Jayaprada, to receive the memento.
All Jayas.
Jayashree, Jaya, Jayaprada.
Here is backed by all Jayas.
All Jayas.
Yeah, Jaya, like, sir, Dhanraj sir, please. So for all of your information, now we are in the final session. See, so far more than 110 online presentations were done by our delegates who have participated in this conference. And every year in the conference, the Society of Life Sciences used to give the best poster presentation award and also the best oral presentation award. In that aspect, the best poster offline, here the presentation goes to Mr. V. Amarnath, MSc, Research Scholar, PG and Research Department of Zoology, Woolwich College. I request Dr. Shvesh Pratap Singh to hand over the certificate. Right. No, let him get it. And for the online poster presentation, there were so many participants. It goes to Ms. Swati Yadav, Department of Zoology, School of Social Life Sciences, Baba Saheb Ambedkar University, Lucknow. And it is in the offline, we will send it by post. And one more, we will send it by post. And then there is an online oral presentation as recommended by Dr. Vijayalakshmi Krishnan. It goes to Sagaya Singh, the state consultant entomologist. Sakya Singh, he goes to that person. Yes, Jharkhand. So these are the posts. It will be sent by post to that particular address. Or else, sir, why can't you receive that award? Okay. Oh, great. Sakya Singh.
Sakya has been like my It is all unbiased. I was not there. I fully trusted on you.
One more thing is that the first keynote speaker is Dr. Kalpana Barua. Though she is not here, she created a very interesting booster to the conference, and on behalf of entire of all of us and Syed College, I would like to hand over the memento to Dr. Kalpana Barua through our friend Dr. P.K. Srivatsa. Please. Sir, this is the memento to Dr. Kalpana Barua.
This is for Kalpana.
Right.
And that certificate is for Sakya.
Yes, sir. Certificate put the Okay, whatever. Digital certificate, you can send it to the
Right.
No, sir, hard copy is there, you can take.
Okay.
But Kalpana madam, he is telling the certificate will be sent to her.
No issue. Hard copy.
The most awaited result is that the best oral presentation, there are two people, we cannot deny them, so it has been decided to give two people. The first one is Dr. Rebecca Vaishali J, Guru Nanak College.
Oh, very good. Congratulations.
The other one is equally the best paper, so we are category, it is not first and second, it is same. So it goes to Dr. Rajalakshmi Anbalagan, Research Associate, ICMR, Vector-Biology Research Lab, Central University of Tamil Nadu, Tiruvarur.
Now expecting some postdoc. Congratulations.
Feedback, okay? Sir, anybody would like to give a feedback about this conference? One or two people who are valuers? Yeah, feedback for the Papa. Okay. One second. Because ஒரே நிமிஷம் சார் ஒரே கேண்டிடேட் தான் அந்த சார் பேசுறாங்க 3 o'clock விட்டுருவோம் நான் 4 minutes 5 minutes போங்க ரைட்
Good afternoon, everyone. The conference was very informative, and I'd like to thank everyone who organized this conference. It was very informative, and we have got many networks of scientists. Thank you for my college and my guide, Dr. Ammal, and Joe Gopinath sir. Thank you for bringing us here. It was a wonderful conference.
Dr. Ramanathapuram, those who do not get time, they can give the feedback for our improvement next time.
So next time, maybe all a lot of feedback.
They can send the feedback.
I invite Dr. Dhanraj sir to the very few words.
Please, sir.
After hectic activities, presentation, this thing, that thing, eating, all those things, they have come to the end of this program. And very soon we will be leaving to our own destination. Right. What I would like to say is, I have been honored like anything there. Your principal, and then Dr. Indian, my boss, Dr. Srivatsa,
No boss.
my colleagues, and then my juniors. I am greatly honored to be in midst of you to see this. In my days, then we will speak nothing, because anything we say, it will go against us. Nowadays we see a lot of colleges, universities coming forward for Vector-Borne. Earlier, Vector-Borne mosquito all government job. It's not ours. Now it's totally changed. And then now new system has come. In those days, you give me a mosquito, like post-mortem, we decide that there is a male or female, whether it's married or not, whether the children are there or not, and that it is veterinary person causing infection. All those things we used to do. But now, modern technology has come. You go to one C-sub leg of the mosquito, or dig up the mosquito with the machine there. The machine will, few minutes it will say what type of mosquito, what type, all those things it has come. So, dissecting and telling stories, no more there. So, new technology has come there. So, what I request the students is to do, yes, engage yourself, do a lot of research. That means, you are doing whatever you do in the Vector-Borne is for the humanity. It's for the this government sometimes may not be able to do that. That is your assisting the government by finding new things that can be implemented properly. Now, this new technology has helped made our work very easier. But otherwise, we call it technology. There is a super specialty hospital somewhere there. So a man went for extraction of the decayed root there. So the dentist, the first case for him. So he pulled anesthesia, even pulled. But since the man was keeping his mouth open, he slipped and fell. So immediately it's an emergency case there. So the patient was referred to laryngologist. Laryngologist from here to here, specialty. And then after giving water the object has crossed my specialty. You have to take him to gastrologist. Gastrologist from here to stomach. So there X-ray was taken. So gastrologist says it has crossed my specialty. Go to the next one. From there, enterologist. Go to the specialist enterologist. There again scanning, meaning everything. The object is not found here. You better go to proctologist there. So what proctologist will do? He will put a proctoscope, you know, from the anus he will observe what is there. Then seeing that, they said that, hey man, there's got a teeth growing here. You better go see the dentist. This is the kind of super specialty we are now having there. But it should be different when we do this thing. So, and then certain things you can recommend to the government. Technical advisory committee is there. Many people are in chemistry and biology, you are doing on insecticides and pesticides and other things. But there is a committee, affects committee at the government level. Technical advisory committee. No fellow from South India is occupying that place. Although we conduct so many research papers, everything, so many students are coming forward for vector control research center, but there is no place for everybody. So they should go to the level of the central government. Equal representation of everyone is required there. So I request all the students take up something there. So whatever you have done, for the sake of doing a paper and then submitting and getting a PhD is not your study should bring positive. And you tell the government, I have a plan here, you better implement it. See that there is no malaria, see that there is no dengue. This should come from there. What happens due to bureaucracy if I go and say, this fellow has done many things. If a student comes, if the university comes, if the college comes out and says, yes, our faculty, our students have done like this, then if you can do this, it will be beneficial for the humanity. So that's how you should come up. saying bringing new innovation into the world of vector control. So it's already time. I say goodbye to everyone. Yes, thank you for giving me the opportunity to address the. Thank you.
Thank you so much, sir, for your insightful thoughts.
Thank you for presenting the effortful and dynamic presentation by scholars, researchers, scientists and all. However, we the Vector-Borne officers from the Chennai Corporation facing practical difficulties, the major domestic sources as well as miniature domestic sources, hidden sources and so many sources, unprecedented sources. So next time we have a session to take all this to the public, not only the public, highest learned people and the lay persons, all this IEC and try to clarify the dengue. Thank you. I invite Dr. Ramanathapuram to relay the word of thanks.
கொஞ்சம் சக்கரை இல்லாத டீ கொஞ்சம் காஃபி பாதி சக்கரை போடுங்க பாயசம் வாங்க சார் வாங்க பேசலாம் உங்களுக்கு 5 hours கூட பேசலாம் நீங்க போங்க அப்படிதான்
Welcome Dr. Sarvakumar to relay few words.
Good evening, everybody. I'm Dr. Sarvakumar, Chief Vector-Borne Officer, Greater Chennai Corporation. I learned vector-borne activities for past 30 years in Chennai. That is my facilitation. Chennai is an unplanned city, having a lot of problems. Chennai is a planetary, having means in level 5 to 7.2. Geographers know. I know about the entomologist. So, as an entomologist, I have joined during 1994. So, I am on the verge of retirement. My request to the users, in future, how they are going to take over this challenges in future. So, before 30 years, we are not having pager, cell phone and other technologies. We ourselves, out of our own interest, we played a significant role. We lost our life. Lost our life means I never focused about my family and kids. People are thinking about, even in my room, mosquitoes come. In my dream, mosquitoes will come. Such a experience I am sharing with you. In future, my request is, I observe that excellent students are coming. Beautiful students. But in my age, that time, they will finish the BSc or MSc. MSc is a big qualification. MSc knowledge is a big qualification. Nowadays, almost all the students, they have finished the MSc, PhD like that. But the qualification is not about anything. Just I want to share my difficulties and how we are going to travel in the future. You always think about field activities. If an entomologist saying himself, claiming himself is an entomologist. without knowing what is happening in the field. He is simply signing and sending reports. I never agree. He is an anthropologist. Vector-Borne is a field scientist. Field scientist. They should be very vibrant. At present, I am experiencing the Chennai Corporation Health Department started as a malaria department. That is before 40 years. Later on, it keep on traveling. Recently, not recently, after a few years, we are reporting dengue cases. Dengue cases, we are claiming that no treatment, nothing like that. Maybe, but keep on reporting dengue cases, other cases, that is top-types and leptospirosis, add on to this list. So, even until now, people doesn't know what is leptospirosis, what is top-types. Both of them are zoonotic diseases. You entomologists know about it. In future, any outbreak occur in your states, how we are going to tackle it? It will be a challenging task. Before that, in this forum, in this conference, I want to convey, please, as an entomologist, you should keep a vigil on not only vector-borne diseases, zoonotic diseases also coming. I am not diverting. This is the conference is about vector-borne diseases. I am saying about this. This is the part of entomologist responsibility. So in future, you are having a huge burden of tackling the vector-borne as well as zoonotic diseases. So in future, you are the backbone of our country. You are the backbone of your state. You are the backbone to neglect the death. Why I am insisting now? So many thousands dengue cases may reported, nothing. Any death occurs. Any death occurs. As an entomologist, we should take responsibility. Even terrorists take responsibility for this blast. I take responsibility. Why not we? So, we entomologists, we should take responsibility if anything happens like that. It should not happen. One. Another thing is, my kind advice. I have traveled more than 30 years in this field. And I read so many thousands of literatures, research papers from 2 decades. Everything. I don't want to expose myself, but my kind advice is the younger birding entomologist should take the stakeholders' responsibility. In future, the country's vector-borne disease is in your hand. You are the stakeholders. We, among the world community, we should not let our pride down. But one day, government will give preferences, privileges, priority this. Until that, we have to prove ourselves. We have the backbone. See, vector-borne and zoonotic diseases, so many death occurs. But there is no accountability, I tell you, throughout India. So, in a few states, leptospirosis is not in the notifiable disease case list. Kerala notified that leptospirosis is a notifiable disease list. Later on, Tamil Nadu claim leptospirosis is notifiable disease. See, notifiable disease itself will create death. Simply I am telling you, it will create death. Recently only Tamil Nadu declared that leptospirosis is a notifiable disease. You think about developing states. I don't want to say the state name anyhow. In future, your entomologist requirement is more. Another thing I want to share with you. Recently, WHO declared one small thing. We are having in our mind that one doctor for 1,000 population, one doctor requirement is there. Who created this? Who created? Who formed it? Nothing. Recently I read in a paper that it is fake. The doctor community itself create such a rumor stating that our country needs one doctor for 1,000 population. You imagine what about entomologists? You imagine what about entomologists? See, for example, Chennai is having 80 lakh population. For 1 lakh population, I am calculating one entomologist per lakh. I am not having 80. See, just imagine. I am not having only 20 entomologists. I am running over. So just I want to conclude my thing. In this forum, entomologists are having a big future. You are need of the country. You are the need of the country. In future, any outbreak occurs. No medically studied doctor they won't encounter it. They will treat, but before treatment, you are the fellows to prevent it, to save lots of economic things for our country. So don't feel inferior about yourself. This is my take-home message. Don't feel inferior that I am an entomologist. You are the number one outstanding stakeholder of the disease prevention in our country. Please, I want to share this through this conference. May God bless you.
Thank you, sir. Thank you for sharing your wonderful experience. I invite Dr. Ramanathapuram to deliver the word of thanks.
So very good afternoon, Honorable here. So I am very happy to deliver the vote of thanks for this 3-day international conference. So on life sciences for human sustainability with special emphasis on control of vector-borne disease, so type 3 mode. So organized by PG Department of Microbiology, Ammal Science College, in association with Society of Life Sciences, Satna, Madhya Pradesh, and the Absolute Human Care Foundation, New Delhi. So first of all, I would like to thank the Ammal Trust and Society of Life Sciences and the Absolute Human Care Foundation. So because without this association, we cannot conduct this program here. So next I would like to thank Dr. Syed Director SMS, and Dr. P. Pradeep Kestivastava, Director of Absolute Human Care Foundation. So these two people are the main for conducting this event here. So thank you, sir. So next I would like to thank our correspondent, Mr. A. Chandra Abdullah, and Mrs. A. Rajati Abdullah, and Dr. Fatima Sanas. So they are very supportive very encouraged for any event. So thank you. So they are not here. So they are very busy. So they are not coming here. So next I would like to thank our active and dynamic principal, Dr. P. Balakrishnan. So without him we cannot conduct any event here. So whatever we ask him, so he will arrange any arrangement. So his support is more to conduct all the events. So thank you, sir. So next I would like to thank our former Chief Vector Control Officer, Dr. P. Tanraj, and Dr. S. Lokumar, Chief Vector Control Officer. So for his sharing in the knowledge of Vector-Borne disease. So thank you, sir. So next I would like to thank all the chairpersons for judging all the sessions. So thank you all. So next I would like to thank all the delegates and the participants. Without you, so you cannot conduct any program. So thank you all. So next I would like to thank the national coordinators, Dr. N.G. Ramanathapuram and Dr. J. Jayanti. So these two people are the main pillar for this conference. So without these people, I cannot conduct any program. So before July 22, I don't know Dr. N.G. Ramanathapuram. So after we conduct. We plan to conduct a conference. We have discussed a lot, nearly 90 days, I think so. So any doubt, I call him. So immediately he pick up my call. So even single ring. So he cleared all the doubts and he gave me a lot of things. So before accepting this event, I was very scared. So how to conduct this program? So because I didn't conduct any program like this. So thank you, sir. Thank you so much. Thank you, ma'am. So next I would like to thank all the executive members and PhD scholars, local advisory committee members, national advisory members, international advisory members. So thank you. So finally I would like to thank all the delegates, all the presenters. So I also congratulate all the winners. So thank you all. Thank you.
For everyone, certificate is ready. You can collect your certificate, have your tea. I will be back after sending our chief guest. I will be back. We will have discussion and then I will tell you what has to be done. Okay? Oh, 5 minutes. I will go and drop him. Sir, come, sir. Thank you. It's our custom duty to send up chief guest.
Tell